Hello everybody. Welcome to the Atto Fridays. It's a pleasure for me to introduce this week's speaker, who is Dr. Hanieh Fattahi. She studied applied physics at Sharif University of Technology in Tehran and received her PhD in physics at Ludwig Maximilian University of Munich in 2015. She was a recipient of the Minerva Fast Track Scholarship of the Max Planck Society in 2015 and was elected as a member of the Schumann Colleague in 2017. And she has been a visiting scientist in the chemistry department of Harvard University and Oxford University. And since 2020, she has been leading her independent research group at the Max Planck Institute for the Science of Light in Erlangen. And she's also the fellow of the Max Planck Center for Extreme and Quantum Photonics in Ottawa and the Max Planck School of Photonics. And her research focuses on the development of mid-infrared and near-infrared field cycle sources, the development of highly sensitive light detection metrologies, mid-infrared overtones and simulated Raman field resolved spectral microscopy, and also solar lasers. And today she's going to talk about femtosecond molecular fieldoscopy. So welcome to the Atto Fridays, and thank you for accepting our invitation. The floor is yours. We're ready to start.
Thank you very much, Clara, for this very nice introduction. So I just try to share my screen first. Let me see. So you see the screen nicely, right?
Yes, it's okay.
Okay. So then I think I should say good afternoon, good evening, good morning, everyone, whoever is interested in watching this talk. So as Clara just introduced me, I'm Hanieh Fattahi, and I have a research group at Max Planck Institute for the Science of Light. So today I would like to talk to you about femtosecond fieldoscopy. So I'm pretty sure you haven't heard the word fieldoscopy before. It's a word that we in my group, we tossed in this word. Just let me see. Yeah, perfect. Just a little bit about the general vision of my group. So we are based in Erlangen at the Max Planck Institute for the Science of Light. Here you can see the very beautiful building that we have. And then the research in my group is focused on two main, let's say, subjects. So we are very much active in sensing, and we also recently started a branch of research on sustainable solutions. So I will talk about all these topics today, one by one. And then for the sustainable solutions, I just give a very brief overview of what we have in the pipeline and what we are working on currently. So I start with talking about the femtosecond molecular fieldoscopy. So this forms the core of the research in my group. And to motivate you that what this research address and what is the question that we try to solve, then I start with the fact that if you would like to do label-free microscopy of soft matter and biological samples. So where we are and how we can contribute more in this field. So this is a very nicely recognized field, right? Microscopy of soft matter. And this is, I think, the Nobel Prize of 2014, which went to several microscopy techniques like STED, PALM, SEAM, and so on. And with this kind of techniques, you can make images of biological samples with a resolution down to currently 10 nanometer. So how is it possible? Because if you know that the light, if you focus it, you are limited by the Abbe's diffraction limit of light, which is around 200 nanometer. So this super resolution microscopy has been made possible by playing with the space and time dimension. And what scientists do to do this kind of imaging, they add fluorescence molecule or a dye to the sample that they would like to image, and they just gather the light which is emitted by these labels. So it's a beautiful technique. It's opening up many new possibilities for imaging of soft matter and in the field of biology. But these labels, they are a bit tricky because they are mostly very big and they affect the dynamic of the sample that you are looking at. And they are also toxic and hostile to the biological sample. So is there any way to avoid them? Yeah, of course, there is a way. And the technique has been called label-free spectral microscopy, and it has been around for several decades now, I would say. So what is the core of this technique? The idea is instead of labeling the molecules that you would like to see in your sample, what about looking at the absorption or emission of the molecules that you have in your sample? So as you know, there are some fingerprint, every molecule or chemical band has some fingerprint at the mid-infrared spectral range, which are called fundamental vibrations, or they have a unique response at overtone or Raman at the near-infrared spectral range. So then it's like that each of us have a fingerprint, then you can find the fingerprint of the molecule, you look at the absorption or emission at infrared or Raman, and then make an image based on that. So this is a beautiful technique, but of course then you are limited by the spatial resolution because you are forced by nature to excite your molecules at the frequencies which is dictated to you by the nature. And then you see that the wavelengths are pretty long, so it makes what it results, then you are going to have images with very poor spatial resolution, or you can go to the near infrared and you can never really go down to very high spatial resolution. But it's a beautiful technique. They are non-destructive. They don't disturb the sample that you are looking at. There is no toxicity, right? And you also gain chemical information. For example, in the type of imaging that you do with labeling, you never know what kind of, what is the constituent of this sample that you are looking at. But with the Raman or infrared, let's say, imaging, you get information about the chemical information of your sample. So what we are focused on, we are focused to turn this red cross to a green tick. This is what we do with a technique that we call femtosecond molecular fieldoscopy. So fieldoscopy is a word that we made in analogy with microscopy and spectroscopy. And by that, we mean that we measure the electric field of light to gather information. So the idea behind it is very simple. So let's say that you are forced by nature to have your excitation pulses at the near infrared or mid infrared, where the resonances of the molecules or chemical bands are. So what about having a probe pulse, which is at much higher frequencies, and somehow use this probe pulse to make high resolution images? Because if you are at a higher frequency, then the diffraction limit of light is smaller, so you should be able to probe smaller portions of the space. So if I just visualize it for you very simply, let's say you have a cell. So let's say the mammalian cell are about between 5 to 10 micron, right? And then you just shine your excitation pulses to this cell. And it's very good to use a short pulse because short laser pulse means a broadband spectrum. And then you can be in resonance with all the, let's say, molecular vibrations which are in your cell, right? And if you look at the transmitted light, then you are going to see the main excitation pulse and the molecular response, which shows up at the trailing edge of your pulse. So this is, in principle, the time domain picture of absorption. So most of the time, if you have a broadband spectrum and you look at the absorption, you see that some frequencies, the intensity at some frequencies is absorbed and it's smaller. But in the time domain, the absorption means delayed re-emission, which is this molecular response that you can see here. So if you find a way to read this electric field of light, then you have access to all the information of the molecules that were in your cell and absorb the light at these very distinct frequencies. Or you have the fingerprint of your cell. So now if you turn on your probe pulse, which is at higher frequency, then it has a smaller diffraction limit, and you can specially gate the information of the irradiated area. So then the question is that, okay, then how I just gain information about the electric field of light? Just remember that we are talking about near infrared. These are the these are the very high frequencies. So you are near petahertz, at the near petahertz frequencies. So then let's say, what about using the same pulse to read the electric field of light? So this is the concept of fieldoscopy that we are developing in my group. Now you see also the relevance of this technique to ultra-short pulse generation and attosecond physics, right? Because you want to now look at the electric field of light. This has been the task of attosecond streaking, for which you have to put the sample in vacuum and you can never do the biology. So now you have to find a way to read the electric field of light at near infrared and make these high resolution images. So what does it give you? Now you see that you should be given very high detection sensitivity because here, and then high dynamic range, because now here the excitation pulse is separated from your molecular response, right? And now you have a kind of background free signal. And of course, you are supposed to get a high spatial resolution because you are temporarily gating your excited area. And because we are using ultra-short pulses, then you get the simultaneous coverage over the entire molecular vibrations in your sample. So it should be a very fast measurement also. So if you do the numbers very well, you see that if you design this system nicely, you can really break the Abbey's diffraction limit. So this is the current state of the art of Raman microscopy. And you see that if you design the system properly, you should be able to get sub 200 nanometer spatial resolution images. So, but everything now is down to the question that how to measure the electric field of light at these very high frequencies. I just show you this very nice informative curve that what kind of information you get when you measure the field itself instead of looking at the intensity of the light. So the panel B is always the typical response that we can get in our measurements, right? If you do frog, X-frog, or any kind of characterization technique. But if you have access to the electric field of light, then you see that you can follow the subcycle dynamic in the matter, how the field, how each cycle of the field interacted with the matter. And you get the complete phase and amplitude information. So during the last decade, there has been several techniques that has been developed all over the world. We have this review paper about it that you can have a look. So here you can see the stars that show the distribution of these novel techniques, field resolve, let's say, technique in terms of energy and the bandwidth. And you see that there are quite some of them, and they can resolve field at very high frequencies. And the beauty of these techniques is that they are operating in ambient air. And ambient air is important because you are not limited to vacuum like attosecond streaking, and you can take things, you can look at things that they can operate only out of vacuum. So the technique that we contributed, and among these several available techniques, is based on very similar to electro-optic sampling if you have a background in terahertz, let's say spectroscopy. And I just very briefly explain how it works, but feel free to look at these cited publications for more information. So how the technique that we developed work. So let's say that you have these very short probe pulses and you would like to measure the electric field of these excitation pulses. So in this panel, I'm just showing you how everything looks like in the frequency domain. So this is the spectrum of the excitation pulses, a broadband pulse at around 1.2 micrometer. And then this is the spectrum of my probe pulses at 300, let's say 50 nanometer. So what I need to do, I need to send these probe pulses and excitation pulses to a nonlinear crystal to generate a correlation signal. And then this correlation signal has to be in a way that whatever correlation I get from this nonlinear crystal, it has to be proportional to the electric field of the excitation pulse, right? And then afterwards, then based on this proportionality, I should be able to just move the probe pulse step by step, like a pump probe measurement on the excitation pulse, and resolve and let's say reconstruct the excitation pulses. So what happens in this nonlinear crystal? The process is actually very simple. In the nonlinear crystal, I just get some frequency between the excitation pulses and the probe pulses. And because everything is so broadband, you see that the sum frequency generation is spectrally overlapped with the probe pulses. And if you look very carefully, you see that there is a very small area that the probe and sum frequency generation spectra are overlapped. So what we do, we in principle just filter this overlapped area, and we look at the polarization rotation of this spectrally overlapped part. And if you do a little bit of math, you see that this part, the polarization rotation of this part, is proportional to the electric field of light. And this way we can resolve the field of the excitation pulses down to very high frequencies. So, but as you see, then to do this kind of measurements, you need ultra short pulses, two ultra short pulses, and they have to be temporally synchronized, right? Because you don't want to have any jitter, temporal jitter between the excitation and probe pulses. And you would like the excitation pulses to be carrier envelope phase stable. Because if shot to shot the excitation, the CEP of the excitation pulse moves, then if you do a multi-shot measurement, everything is going to average out to zero. So this is a type of, let's say, measurement that you can do with this detection technique if you combine it with spectroscopy. So you can just follow this attached, cited paper that we just recently published in Nature Photonics, that we show that if you have a, let's say, liquid sample, and you just shine your excitation pulses at the resonance to this, to the molecules in the liquid, and resolve the electric field of the light afterwards, then you can detect the response of the molecules very nicely. So the typical measurement looks like this, that you have the excitation pulses, and then you get the liquid response, which is up to 1 to 2 picosecond. And you can see that it exponentially decays. And then because of the very high sensitivity of the system, you are also able to resolve the response of the atmospheric gas molecule that you have in your system. So you somehow, by field measurement, you can distinguish between the different phases of the matter as well. So now the question is that we need these very short pulses, and we would like to do this kind of measurement, and we would like to have these pulses at the laser source at a very high repetition rate. So what is the reason behind it? Because, of course, if you would like to do spectroscopy or imaging, then you would like to scan fast, right? So if you want to make an image, you don't want to wait for making an image for 24 hours. And there is also this 1/f noise law that if your system operates at high repetition rate, let's say megahertz, then the noise of the system approaches to the shot noise of the photons, right? Because then you get rid of all these vibrational and slow noises in your environment. But this also brings so many challenges, because if you have to use a system, ultra short pulses at high repetition rate, then you have to think, okay, how this soft tissue is going to interact with these high average power, high peak intensity pulses. Is the soft tissue, is it going to survive this condition or not? That we aim to image soft tissue at the end, and we don't want to damage them. So high average power is also challenging. You have to deal with thermal issues all the time. And also, these kind of systems, they have the high repetition rate lasers. They have usually lower peak power. So the technique that you can use to generate ultra short pulses are very limited. So the technique that we chose to generate what we require to do this kind of field measurements is based on fibers. So we chose these single ring fibers, and based on the dispersion, let's say, engineering, then we could run this fiber in two different regimes for spectral broadening. So if you feel a gas with a specific dispersion, then you can tune the total dispersion of the fiber and the gas to a normal regime. And here, then you can get a normal self-phase modulation type of spectral broadening. And here I show everything in the frequency domain and the counterpart in the time domain. And if you just play with the gas that you feel in the fiber, then you can tune the dispersion to anomalous regime. And this is somehow where the magic happens, because then you can have up to 1 octave spectral broadening, as you can see here. And because of the tuning of the dispersion and so on, in the time domain, your pulses also get compressed. So you are going to have a soliton self-compression and spectral broadening at the same time. So this is very nice, because then you don't have to think all the time, okay, how I can compress now this very more than 1 octave spectral bandwidth. So how we did it, I just walk you through the experiment. So we start with a conventional laser. This is a carbide laser which operates at 1 megahertz, delivering 250 femtosecond pulses at 20 microjoule of energy. Then we focus these pulses to a 50 centimeter long single ring holocore fiber, as I showed in the previous slide. And at this stage, we just fill the holocore fiber with 14 bar of argon. So at this part, we have the spectral broadening based on cell-phase modulation. So we have to use chirp mirrors to compress for the accumulated GDD during this process. So then after the chirp mirror compressor, we could successfully compress the pulses to around 2730 femtosecond. Here you can see the typical cell-phase modulation spectrum that we can get from such fibers. So after that, we just send these pulses to the second fiber stage, which is exactly the similar fiber, but this time we just fill the fiber with 15 bars of helium. And that's where the magic of the soliton self-compression happens. And the pulses are compressed at the output of the fiber. But of course, we have the window here of the gas cell, and the pulse propagates a bit in the air and can get stretched. So we use a couple of reflections on chirp mirrors for pre-compensation. After this stage, then you can see that we can broaden the spectrum at very high repetition rate, with more than like an octave spanning bandwidth. And after 3-4 reflections on chirp mirrors, we have the pulse duration of 4.5 femtosecond. So the pulses at the output of the fiber actually are even shorter. They approach the single cycle regime, but the bandwidth of chirp mirrors couldn't support the compression of the pulses to their actual Fourier transform limit. So these 1.5 cycle pulses are the workhorse of somehow the system. We use them to generate the CEP, stable excitation pulses, that we need to get this spectroscopic information from matter, for example. So how we do this excitation pulse generation? It's very important for us to have a very good carrier envelope phase stability. And the process that we use intrapulse difference frequency generation. Because the bandwidth of the pulse after the second fiber is very huge, then if you focus them in a nonlinear crystal, in this case, a BIBO crystal, then different spectral components within the pump, they start to mix with each other. And you can get the down-converted, let's say, pulse at the longer wavelengths. And because you have this intrapulse process, then the generated pulse is CP stable. Then we use a small portion of these probe pulses as a small portion of these 4.5 femtosecond pulses as a probe pulse, and we send everything to our field detector. So here you can see a typical measurement that we use for pulse characterization. So this is the electric field of the generated DFG pulses, the intrapulse difference frequency generated pulses. Then you can see that we can even have a very high, let's say, detection dynamic range and detection sensitivity, that if we attenuate the field by orders of magnitude, we are still able to see the electric field with our detection technique. So, and you can see the spectrum of the excitation pulses, which is broadband and can cover many overtone and combination bands of important molecules, let's say, and also the detection sensitivity and dynamic range. Okay, so if I bring you back to this figure that I showed in the beginning of my talk, how we are contributing to the field, then you can see that we just push the sensitivity of this kind of detection technique by orders of magnitude to femtojoule pulse energy. Now, if you just add some molecules, sample, liquid, I don't know, soft matter, then to the beam path and measure the electric field, this is how a typical measurement looks like. So then you see that there is an excitation pulse, there are some molecular response, and which are ringing for quite a long time, depending on how far you just scan the delay and measure them. So this panel, this figure A, shows you a typical measurement that has been done. So you always look at averaged response of the sample with your spectrometer. So if I Fourier transform this electric field in the time domain, then you can see the response, which is this gray spectrum. And you can see that you notice that there are some absorptions, but it's really hard to interpret the data. This is the typical picture that you get from your spectrometer when you want to do, let's say, spectroscopic measurement. But now that you have access to the field, you have the possibility to gate your measurement. So now in panel A1, I show how the electric field of light looks like if you gate it from around 0.2 picosecond to 1 picosecond. So this area. So this is a typical response of liquid that if you have in your sample. And if you Fourier transform this, then you see this very nice blue curve, which shows very well the broadband absorption of the liquid that you have in your beam path. And if you temporally gate the response at the longer time scale from 1 to 6, from 2 to 6 picosecond, for example, as it's shown in panel A2, then you can get the response of the gas which is in your environment. And if you Fourier transform it, you get this, let's say, red curve, which shows very distinct spectrum in the gas phase. So we benchmarked this system. We looked at several alcohols and also um, several types of liquid at different phases of matter, like like a vapor or in the liquid form. And we show that we can resolve the very well-known absorption bands in these kind of alcohols and matter, and our data matches very well with the HyTrend database. So to show that the system works very beautifully to recover the very sensitive spectroscopic information of different samples. So now that we have this very beautiful sensitivity, let's say dynamic range, and we can get the electric field response, we can get access to the electric field response of molecules at very high frequencies. What about doing nonlinear kind of spectroscopy with this kind of system? So this is the work that we tried. So here you can see our concept for the stimulated Raman fieldoscopy. Everything is the same. So we have an excitation pulse, which is broadband and is focused to our sample. And then to do the Raman, then you need to turn on another, let's say, laser pulse on your sample, and that's the narrowband Raman pump, right? And if the difference of the frequency between your Stokes and Raman are in resonance with the bands, the chemical bands of your molecule, then you can have the Raman gain, let's say, in your Stokes pulses. And then if you look at this picture in the time domain, then again, the same logic, right? So you have your excitation pulses and the molecular fingerprint, which is delayed in the time domain. There is one more beautiful thing in this response is that you can, if you manage to measure the electric field, you access to the Raman response of your molecule and also whatever nonlinear process that happens during this process, during this interaction of the Raman pump and Stokes pulses on your sample. So this is how the typical, let's say, setup looks like. It's very similar to the previous one, but now here we also add the Raman pump to the system. And we have the sample in a kind of a liquid jet that can circulate the liquid. And the light now this time is focused to just get this nonlinearity in the sample. To get a very, let's say, good signal to noise ratio, then you have to use the lock-in system. And for the Raman process, the beauty is that we can modulate our signal with the AOM at very high, let's say, frequencies. So this is how our modulation system looks like. So the Raman pump is modulated at 500 kilohertz. And the Stokes pulses, which are like the excitation pulses, are not modulated and they are showing up at 1 megahertz repetition rate, which is the repetition rate of the laser. So then after interaction with the molecules, you only have the Raman process happening where when there is a presence of the pump and Stokes pulses. And this way we can demodulate the whole thing at the 500 kilohertz, let's say, rate. And as you see, then this gives us the possibility to get a completely background-free detection of whatever process happens during this nonlinear process, the nonlinear interaction. So this is a typical response that we recently got from the acetonite trial in a 400 micrometer thick liquid sample. And you can see that if you temporarily gate again your response and only look at the initial portion of your pulse, you see that we can see that the Stokes pulses are amplified. So this is based on the Kerr instability and amplification based on Kerr instability, and that now we can resolve its electric field and we can show that this broadband process is happening. And if you just look at the later time scale, right, like this molecular fingerprint, let's say, and you Fourier transform it, then you can resolve very beautifully the Raman bands of the Raman active bands of the acetonitrile. So if you look closely to this curve, you also see this very small, let's say, portions of energy that they are appearing at the off resonances, that they are very nicely overlapped with the response of the atmospheric water molecule. So this again shows the sensitivity of the system, that we can, with one measurement, we have access to the information of whatever is in the environment. We have access to the nonlinear response, the full nonlinear response within this interaction, if it's a four-wave mixing or Kerr instability, and we can have access to the Raman response, broadband Raman response of the molecular system. Okay, so we extended this technique also. We are extending it now towards optical coherent tomography, because if you have a sample with different, let's say, variations of refractive index, then this variation in refractive index just translates into the delay, temporal delay, right? For the light that passes through this modulated, let's say, area. And if one can measure the electric field, then you can calculate back and learn about the thickness of these changes in the refractive index and so on. This is the measurement that we did, for example, that we characterized that we are sensitive to measure refractive index changes of a sample with a thickness of about 234 micron, 250 micron, and we can easily recognize also all the spectroscopic information that happens in these individual areas with different refractive indexes. So this holds promise to do optical coherent tomography with chemical sensitivity. Now, you might think that, okay, how can we accelerate, let's say, the measurement, this kind of measurement? And if you look at closely of what I explained, you see that we are only interested in this molecular response or molecular fingerprint in the time domain. And this is a sparse response in the frequency domain, because there are only very specific frequencies that were absorbed in the matter. So then in principle, you should be able to use kind of compressed sensing processes for the fast reconstruction of the electric field with sampling fewer, let's say, data points. So this is what we showed, and it's recently published in Ultrafast Science if you would like to look at more details. So we developed a very similar system. And what is important in this kind of measurements is that your sampling has to be random. So you have to be able to random your points in the time domain to sample them randomly. Otherwise, the compressed sensing won't work, right? So to do this random, let's say, sampling, we use Dazzler in the collaboration with the Fast Light company, which gave us also very fast scanning range. And you can really shot to shot move the laser pulse. So let's say that if you would like to measure such electric field, then the probe pulse with the Dazzler can move in random positions. So if you do this type of field result technique, then you see that with much fewer data points, we are able to reconstruct the spectrum of the absorbed light, right? So this is a typical, let's say, measurement that you need 2023 data points, and then you can reduce it by orders of magnitude and still recover the electric field of the light and the absorbed frequencies. And then we show that you can really resolve the response or the frequencies below the Nyquist limit, and you can really overcome this barrier. So now I motivated my talk with microscopy, but the whole time I've been talking about spectroscopy. But if you think about it, microscopy is in principle a raster scanned spectroscopy technique. And in the beginning of the talk, I mentioned that one of the challenges of using ultra-short pulses for imaging of soft matter is that if if the photo damage is an issue and if they are suitable for this kind of experiment. So this is the study that we performed with similar pulses that we need for such a microscope. And in this study, which is an in vivo study, so it's very valuable, let's say, in terms of biological understanding and application, we irradiated the the central nervous system of zebrafish larva with different pulses at different wavelengths and different repetition rate. And we just measured and identified at which peak intensity the damage threshold occurs. And if you look at this curve, then you can see that, of course, the higher frequencies are very damaging for biological applications because you have really direct absorptions. But as you move to the near infrared spectral range, you can really use very high peak intensities without destroying or damaging the cells. So this put us in a very good situation because our excitation pulses are designed for near infrared, short wavelengths infrared. So, and then they have to be intense. So it's very safe for biological application. And our probe pulses are at UV, but the good point is that we don't need very intense probe pulses to do this kind of imaging. And the intensity that we need for the probe pulses is well below the peak intensity that we measured for biological damage, let's say. Okay, so then we put everything in the microscope geometry. Here you can see the fiber spectral broadening stages. And then everything is the excitation pulses and the probe pulses are focused in a kind of reflective objective on the sample. And then we have the nonlinear crystal, which we use for the detection attached to the sample. And then we measure the polarization rotation afterwards. Here you can see how we win in terms of a spatial resolution. The excitation pulses have a bigger diffraction limit, and the probe pulses they can specially gate, let's say, the irradiated area. So to prove somehow the resolution of our system, I mean, if you have a microscope, you just buy one of these kind of resolution target from Tor Labs, and you just see how far you can see it. But we needed a kind of chemically active or a kind of resolution target that has interaction with our excitation pulses and absorbs our pulses. For that, we teamed up with a group of Ilka Kriegel in Italy, and we they made this kind of, let's say, sample for us based on indium tin oxide nanoparticle. And then we showed that we can resolve the response very nicely with our field result technique down to monolayer. Then we use this as a resolution target. Here you can see that the ITO is placed on the nonlinear crystal. And well, this is the picture of the setup, how it looks in the lab. You see that it's not very big, so it fits to one optical table. And right, then you can just raster scan point by point, measure the electric field of light. Here I show you the image that it's made from the corner of this structure and make the image of your ITO structure. And in each pixel, you have the full chemical information of the sample that you imaged. So this is a very good news because now you can do hyperspectral microscopy with attosecond, let's say, precision in the time domain. Where we would like to go, of course, we would like to look at some very exotic, let's say, molecules. These are the neurotransmitters, which are the molecules that that the neurons are releasing when they want to communicate to each other. So they are very small, so it's almost impossible to label them. And the neural gap is very small, so this area is around 200 nanometer. So then our technique can play a very good role for detecting these neurotransmitters, which are the origin of many diseases or, let's say, many emotions that we have. So we already have done some preliminary measurement, and we could see that we are sensitive to many of these groups of neurotransmitters. Okay, so with this, I just would like to talk to you a little bit about our work on pushing the, let's say, the generation of infrared pulses for gas spectroscopy, and afterwards I give you some words on the solar lasers. So this is also a very recent result that we have, and in this, let's say, work, we combined ytterbium YAG-thin disk laser technology that you can see here with optical parametric oscillators to generate broadband pulses, high power, bright femtosecond broadband pulses at near mid-infrared and short wavelength infrared. So ytterbium YAG-thin disk lasers have been somehow very much in attention. And this is because you can scale their energy and average power simultaneously. So they can be a gate for the generation of energetic, powerful pulses at longer wavelengths. So with a very careful design of the cavity, with the OPO cavity, we could get OPO pulses, let's say optical parametric oscillator pulses at 2 micron, which were very nicely modulated. Here I show you the spectrum of these pulses. You can see that you can tune them from 2 micron to 1.9 micron. And you can see that they have several overlap with the overtone of several atmospheric molecules. And somehow this is somehow a path to just scale the energy and average power of these kind of systems. And on top of that, this broadband modulation that we have on the signal will increase the detection sensitivity hugely as we can use the, let's say, fast detection techniques. Okay, so then I would like to wrap up my talk to just give you a little bit of impression on our attempts for developing sustainable solutions. So we are very much focused on developing solar lasers. Solar lasers are in principle lasers which are pumped by the energy of light, and they can have several applications like in energy, in communication, and in developing sustainable, let's say, energy loop like magic. It's interesting to know that the first solar laser was already shown in 1966, very soon after the realization of the laser. And you had to just concentrate the solar light to your gain medium and track the sun. And Jung could get up to 1 watt of CW laser output with this geometry. But if you look at over the years, you see that in the early years, it has been shown that lasers can be pumped by sun. But very soon, people learned that they can use other lasers to pump their laser, right? and it's much easier, of course. So then we didn't have so much development, but in recent years, there has been many work addressing this topic. And that's because of the energy crisis that we face at the moment in the world. And if you look at the somehow the state of the art, then you see that in terms of the generated power, we are very much saturated. So the light has to get, we have to focus the solar light very tightly. We have to track the sun. Then you have to cool down the gain medium, right? And then at the end, you end up with hundreds of watts of, let's say, output power. So if you would like to get out of this state of the art, then really a new idea or a new concept is needed. So you can also think about using different gain media, right? But it's also very tricky. So this is the solar spectrum. You have many frequencies. And if, for example, you use these ytterbium YAG lasers, which are very nice, then you see that you just absorb very small portion of the spectrum and emit at a very specific wavelengths. You can look at other gain media, for example, titanium sapphire. It has a very nice absorption at the peak of the sun. It also emits a broadband, let's say, spectrum. But when you look very closely, you see that this gain medium is a three-level system. So it's really hard to reach the threshold and have the laser lace. So then there are several questions. So how to increase the conversion efficiency of the solar lasers, how to cool them down, how to increase the collection, the efficiency of the solar light collection, and how to change the state of the art somehow. So these are the two projects that I would like to advertise here. So the first one is Apache. This is an EU Pathfinder project, which my group is part of it. So it's a consortium of 5 different universities and institutes all over the Europe. And the idea behind the Apache is that to be inspired by nature. So how to be inspired by nature? Let's say if you look at the photosynthesis process that happens, it is very efficient. So the plants and the leaves, they can absorb light very nicely and convert it to energy. And if you look very closely, you see this is because they have these antenna complexes or these big molecular aggregates that they just absorb the sunlight and then they transfer the energy to their center, which is which is called the reaction center. And then here some chemical processes happens and provides the energy for the plants. So in Apache, we are very inspired by the photosynthesis process. At the moment, we are developing these antenna complexes with our collaborators, and which should just harvest the sunlight very nicely. And then the energy is transferred to the center where we have our laser gain medium. And this way, we are aiming to develop low threshold solar lasers that they don't need the efficient collection of light, focusing of the solar light, and so on. In the other project that I would like to advertise here, we are looking at solving the issue of the cooling in solid state lasers by combining, let's say, ytterbium YAG thin disk lasers with radiative cooling. And we hope that with our new design, we reach the population inversion and we can have a radiatively cooled solar laser pumped by solar light. Okay, so with this, I would like to wrap up my talk. There are several people that I would like to specifically mention related to the talk that I just presented to you. So especially the 2 PhD students on this project, Anshit, who developed the system and showed the spectroscopy application of the field resolved spectroscopy at near petahertz frequencies. Andreas, who developed the microscope and made the first images based on this concept. Kilian here, who demonstrated the stimulated Raman fieldoscopy. Soyan, who did the fish measurement, the damage measurement, and she's very active on the microscope. And Michael, who is actively working on the Apache project to realize the solar laser, and Sahil, who is working on radiatively cooled lasers. And of course, all the other team members who are working on the other topics that I didn't cover in this talk. And I would like to thank the funding, right? The funding agencies and the money, the Max Planck Society and the European Research Council. So thank you very much.
Thank you so much for this really interesting talk. It's impressive what one can do nowadays with imaging. I was really not aware. Are there questions, comments? Would anybody like to ask something? Please raise your hands or put your questions in the Q&A tab. I mean, one question I have out of curiosity, because you mentioned that you have been looking at soft matter and that it's really you aim at looking at brains. You're testing this in some kind of animals, and in the future you want to look at neurotransmitters. How far is the current research, and how much are you bringing with regard to the state of the art?
So there has been attempts to look at the neurotransmitters with, to detect, let's say, the concentration of neurotransmitters with Raman imaging.
Mhm.
But then the issue is that these are really small, let's say, molecules. So you need to have a very high sensitivity. So the measurements, they had huge error bars. And then also the area that you want to look at is very small. It's below, let's say, 200 nanometer. And the current Raman techniques couldn't really reach this very specific area, right? So then with our technique, we very much hope to be able to measure the concentration and bring the spatial resolution in to be able to somehow monitor this kind of small molecules. Yeah, and as you say, it's amazing, right? That how field detection of ultra short pulses can play a role in other fields, right? Yeah.
Yes, I mean, I'm positively shocked. I really didn't know a lot of these things. Congratulations, it's great work. Is there anyone in the audience who would like to ask something? Aha, we have Matthias. Yes, Matthias, go ahead. Hello.
Yes.
Hi, Hani.
It's good to
It's been
see
a
you
while.
here.
Yeah, so really cool. Cool stuff. So I was wondering, what is the bandwidth limitation? So what is the highest frequency you can detect?
So in principle, with the technique that we developed or we are using very actively, so your limitation is your probe pulses, right? So because you need a probe pulse which is shorter than the cycle of the pulse that you would like to measure, right? And you can make these probe pulses shorter and shorter. This is the aim that we are following in this new, let's say, ERC funded project. And you can really go down in UV, right? But then at one point, then I think the BBO has the absorption at the BBO starts at 180 nanometer, right? Or 120, I guess. So then you have these very hard limits that you cannot make the probe pulses shorter because they just start to absorb in your nonlinear crystal. And that defines the limit in the frequency that you can detect.
Actually, it's the some frequency that's limited, right? Because
Right,
you
right.
detect it always as at the shorter wavelength even.
Right, right. I mean, you can think that, okay, I put a gas or something, right, that doesn't
Yeah.
absorb. But for us, because we are very much focused on imaging, so we need a nonlinear solid to put our samples on, right? So that somehow the limitation of us for the imaging. But
Well,
if
for the gas, I mean, you need a second order nonlinearity, right?
right, yeah, or you
You
can,
would need to use, well, something
yeah,
complicated with some
yeah,
blind.
yeah. Yeah,
uh,
correct. Right.
I
Yeah.
don't,
Because
yeah,
you lose this, you have a symmetric system also, right? Yeah.
yes,
You have to just
yeah,
modify the concept, right? Yeah.
yeah. What I also really liked was your impressive pulse compression
Mhm.
results.
Thank you.
So this, you use something of the order of 10 watts, is that right?
So the pulses are 20 microjoule at 1 megahertz. So it's like 20 watts, right?
Yeah, and the efficiency is really nice.
Yeah. We
What's
have a
the
very good throughput, right?
why is it so much better than everything we were used to?
Oh, is it better? So I think the
I
whole
think
thing
so.
is that we are using this single ring hollow core fiber. So
Yeah.
I think the whole thing is that we get this soliton self compression, therefore these tens of microjoules of energy, which is a very tough regime, right? It's not inten the energy is not enough to go to capillary, right? And
Right.
then it's damaging the solid core fiber, let's say. But if you stay at this single ring hollow core fiber, then you can get very good nonlinearity, right? And then
Mm.
the soliton self-compression actually helps a lot because if you just compression of these pulses are not easy, right? If they are
Yes,
very,
absolutely.
very sparse, right? Yeah.
Yeah.
So
And
then the magic I think is in the fiber actually.
do you see any problems scaling this to the, say, 100 watt scale?
This is coming. So then for this ERC project, we are starting with a 120 watt source.
Okay.
Yeah. And I know it's important, right? Scaling the average power is important. I mean, it's important for high harmonic generation, ultra short pulse generation. I mean, attosecond physics, it's all important, right? So to
Yeah.
get sounds right.
So I used to work with something of the order of 10 watts, and then thermal issues were not a big deal. And
Mhm.
now with 100 watts, it's really tough.
Mhm. So you are focusing in the fiber or?
So we've used fiber, we've used multipass cells. It's the thermal issues are everywhere.
Yeah, yeah, they are going to show. So what actually there was one thing that we were very much surprised when we did this soliton self-compression because you have a very high average power, right? Then you also, I mean, the spectrum is very broadband. You get spectral components at very high frequencies. And then if your environment is a bit like polluted or you have some organic molecules, then you
Uh-huh.
deposit them, right? So these are really tricky regime. I mean, going to UV and increase the average power. I mean, the thermal stuff and then these contaminations, they have to be very well taken care of, right?
Yeah,
So you
cool.
are doing the 100 watts with the carbide or?
That's active fiber systems. So it's actually
Ah,
200
I
watts.
see. Oh, I see. I see. I see.
Mhm.
Right. That's
Yeah.
also tricky. That's also tricky.
Yes.
Very tricky.
Okay.
Because you don't have also so much pulse energy, right? How
Well,
much?
the pulse energy is 2 millijoule.
2 millijoule. Okay. Okay.
Yeah. So 2 millijoule, 100 kilohertz, and then you have your 200 watts.
Okay. So then I think I'll call you very soon when we
Yeah,
try
please
for the
do.
thermal issues, right? Yeah. Yeah. Yeah.
Okay. Yeah, cool. And then, okay, maybe also a comment on the final part of your talk, the solar laser. I mean, this is very interesting. When I first heard of the idea, I thought this was cool, a cool thing to do. But I mean, economically, is this really the main motivation? I mean, my point is, photovoltaic panels are really cheap
Yeah,
nowadays.
so with the solar laser, somehow the market is in space application. I didn't mention because I just looked and
Uh-huh.
saw that my time is really, I'm just talking beyond the time. So the niche is in space application. So you
Uh-huh.
need somehow a kind of sustainable source of energy for propulsion and also, there are these dark valleys on the moon that you would like to access. So then the solar lasers can, because then these are the parts that the sun never shines, right?
Right.
And these are the parts of the moon that water is there.
Okay.
can not get the energy by photovoltaic cell or something like that. So then we hope that if with solar lasers can play a big role there.
Okay, this is okay. Interesting idea.
You can just somehow transfer the energy by the laser to the yeah to
Okay.
say these valleys and stuff, right? But then the niche is in principle, the application is, I mean, the let's say space community is very excited about it, right? That if we get
Nice. Okay. Cool. Thank you. Let's keep in touch.
yeah, it was great talking to you.
Yeah. See you soon. Bye.
Thank you. Bye bye.
Bye. So are there further questions, comments? For those on YouTube who are watching, I can see there are a few of you. Take good care of yourselves. We thank once more Dr. Fattahi for this really interesting talk. And I'm going to finish the streaming. So stay safe, have a great weekend, and see you in 2 weeks.